Hello to you in Ukraine. I'm Dr. Sharon Aharaoni from the Neuromuscular Clinic, and I'll be talking about Duchenne muscular dystrophy therapies. So this is an overview. As Dr. Kuzminski said before, corticosteroids use in following standard of care clinical guidelines remain the mainstay of optimal care for boys with DMD. The current standard of care does not treat the underlying cause of the dystrophin mutation, and there are different therapeutics approaches in development for DMD that are directed against a wide range of therapeutic targets. And accurate and early genetic diagnosis will become even more important in this era of evolving genetic therapies. So, what are the strategies in the treatment of Duchenne muscular dystrophy? As you see here in the slide, there are different strategies. One strategy is to act against inflammation, to act against fibrosis, there's cell therapy, and the gene therapy. And this table shows you the therapeutic approaches in Duchenne. The first one is dystrophin protein restoration or replacement. This is the exon skipping or the stop codon read-through, gene therapy, gene editing, the anti-inflammation and fibrosis. It's the steroids that you had a talk before. There are novel steroids like the vamorolone. We'll be talking about it. The NF-kappa B inhibitor, the HDAC inhibitor, the gindivostat, tamoxifen, calcium regulation. These are the ryanodine receptors and the calcium homeostasis. There are agents that act against muscle growth and protections. This is the myostatin inhibitors, the eutrophin upregulation, and the stem cells products that act in the mitochondria. These are the antioxidants like the idibenone, cardiac agents, the traditional cardiac drugs, stem cells, and therapeutic approaches that alter blood flow, like the nNOS upregulations and other therapeutic approaches. First, we'll be talking about anti-inflammation and fibrosis. There are alternatives for steroids because, as you know, steroids have a lot of adverse events that we don't want to. There's a drug that is in development now. It's vamorolone, and it resembles prednisolone in chemical structure, and it resembles prednisolone in NF-kappa B inhibition, but it has less side effects. And this drug is being in clinical trials here at Schneider's. Vamorolone is similar to prednisolone. As you can see, vamorolone is here colored in red, and the prednisolone is here in blue. Vamorolone is a first-in-class drug that borrows some activities of steroids, like anti-inflammatory activity via NF-kappa B repression, while adding new activities not seen with corticosteroids, like membrane stabilization activity. Vamorolone hopes to retain the beneficial anti-inflammatory and muscle strengthening aspects of corticosteroids while decreasing some of the undesirable side effects like bone fragility, stunted growth, insulin resistant, mood changes, delay of puberty, and others. These are side effects that we don't want. And vamorolone is given as an oral liquid flavored formulation at home daily. Another drug that is being investigated is the CAT 104. It's from the pharmaceutical company Catabases. It's an oral small molecule investigational drug candidate that inhibits NF-kappa B, a potent promoter of inflammation. And by inhibiting NF-kappa B, it is thought that this agent may reduce muscle damage and increase muscle regeneration in boys with Duchenne without the side effects of steroids. But on October 2020, Catabases announced that the phase 3 Polaris DMD trial in DMD muscular dystrophy did not meet its primary or secondary endpoints. So the trial was stopped. Gingivostat is an histone deacetylase inhibitors that have been shown to activate gene expression and to have a beneficial role in the MDX mouse model. Gingivostat is an HDAC inhibitor that has anti-inflammatory, anti-fibrotic, and regenerative properties, which is in phase 3 study in ambulatory DMD patients. And this trial of Italia Pharmaco is also being studied in children in Schneider Children's Medical Center of Israel. We will not speak about all the strategies, approaches for treatment, but a very important strategy that I want to talk about is the dystrophin protein restoration or replacement. The approaches are the in-frame nonsense mutation read-through. These are treatments that lead to introduction of amino acids at in-frame nonsense mutations like premature stop codons. The exon skipping, these are synthetically modified DNA or RNAs with antisense oligonucleotides that promote exon skipping to restore functional dystrophin protein expression for types of deletion mutation. And then we have the gene therapy, introduction of the functional dystrophin gene directly into muscle fibers to restore sufficient level of expression. And then there's the gene editing. The dystrophin gene or protein is the largest gene and protein in humans. It has 2.4 million base pairs and 79 exons, as you can see here. Now, when the reading frame is read, this deletion is an in-frame mutation, and this is a BMD, a Becker muscular dystrophy, with less dystrophin, but it's still present. When the reading frame is not read, like in here, like seen here, this is an out-of-frame mutation that is seen in Duchenne, and dystrophin is completely absent. In deletions of the dystrophin gene, 13% of them are of exon 51, 80% of them are of exon 53. And this is important for the exon skipping, which I'll be talking later. 20% of the gene D deletions may not be amenable to exon skipping. Many clinically relevant mutations have been identified in Duchenne, such as deletions, duplications, and small mutations, including nonsense mutations. Mutations prevent the creation of a full-length functional dystrophin protein. Most of the mutations, 68%, account for deletions, 11% are duplications, 20% are small mutations, of those 10 to 15% are nonsense mutations. Ataluren, or Translarna, is an orally delivered small molecule designed to selectively induce ribosomal read-through of in-frame premature stop codons to produce full-length dystrophin. Ataluren is given 14 milligrams per kilo and is indicated for the treatment of DMD in ambulatory boys. In these countries, it is indicated above age 2 years of age. In Europe, Iceland, Israel, and other countries. In Brazil, Chile, and South Korea, the indications is for boys above 5 years of age. Ataluren is considered an investigational product in the US and is not FDA approved. Translarna is proposed to restore protein production by enabling ribosomal read-through of mRNA containing an in-frame premature stop codon. As you can see here, this is the ribosome, the amino acid. Here we have a premature stop codon. This is the mRNA. A premature stop codon causes translation to stop prematurely, and Translarna enables complete translation, reading through premature stop codon, and the translation of mRNA continues, as you can see here. Exon skipping or RNA repair. There are two drugs that have been approved by the FDA. One is the Exondys 51, and the other is the Vyondys 53. Exondys 51, or eteplirsen, is for treatment by skipping exon 51 of the DMD gene using PMO chemistry, and that accounts of 13% of DMD patients. Vyondys is for treatment by skipping exon 53 of the DMD gene, and that accounts of 8% of the DMD patients. There are further treatment trials with PMOs directed to other exons. For instance, casimersen for exon 45 that is studied in clinical trial in Schneider as well. Exon 51 skipping is induced by eteplirsen, leading to production of truncated but functional dystrophin protein. And I'll show you how it works. This is the normal dystrophin mRNA, and this is the ribosome that reads the reading frame, and there's normal dystrophin protein. Now, when there's an exon 48 to 50 deletion, it disrupts the reading frame, and so the ribosome has trouble to read the reading frame. And there's, as a consequence, there's an unstable or no dystrophin protein. That's what happens in Duchenne. And when we have a skipping exon 51, it restores the reading frame. And so the ribosome can read it, and there's a shortened but functional dystrophin protein. Now we're going to talk about gene replacement therapy. Gene replacement therapy by viral vectors has been popular in the last years, and the most famous disease that it has been used is the spinal muscular atrophy. Zolgensma is a drug that is developed by viral vector, the AAV9, and the vectors are used for oncology disease and for other rare disease. And what's unique about the AAV vector, as you can see, there are other viral vectors like the retrovirus, lentivirus, herpes, and adenovirus, and the AAV, the adeno-associated vector, is unique because it's non-pathogenic, it's generally non-immunogenic, it has low or non-inflammatory advantages, but its main limitations is that it has a small packaging capacity that limits the range of therapeutic genes that can be used in the system. It has broad tropism. And this is how the AAV capsid and vector is engineered. It has a capsid, which is the shell surrounding and protecting the viral genetic material. The original viral genetic material is taken out and the transgene is being replaced. And for AAV vectors, it is largely the capsid that determines the tropism and transduction profile in different cell types, and research has focused on developing AAV capsids that have unique characteristics. Duchenne is an attractive candidate for gene transfer as it arises from mutations in a single gene, and among the most promising experimental therapies is gene replacement therapy that uses AAV9 vectors. And the challenge to gene replacement therapy for DMD is that the DMD gene is the largest gene in humans. As I said before, at 11.4 kilobase, it is too large to fit into an AAV genome. And as a result, several groups designed microdystrophin genes or mini dystrophin genes, like Pfizer, Sarepta, and Solid, that could fit in this vector. And in Schneider, we just started this trial with Pfizer, and we hope to recruit boys for gene replacement therapy. In the end, there's the gene editing, which is the CRISPR or Cas editing. And if this is the DNA, the scissors here represent the Cas9 or the CRISPR that cuts the DNA. CRISPR can be used to skip one or multiple exons, and it's similar to exon skipping, but CRISPR makes a permanent change in the DNA, so there's no way out, and we have to be careful about it. So, in conclusion, there are many possibilities in the near future for novel therapies that may significantly alter the course of DMD. We still have to follow the standard of care for DMD by treating with steroids and multidisciplinary care. And early and accurate molecular diagnosis is important. And by this, I'll say thank you and goodbye for you all. Thank you for listening.